Welcome to AMiNDR, a podcast where we summarize the latest publications on neurodegenerative disease research so that you can stay up to date with the newest findings. Every month, our team of scientists will sort and organize the titles into themes and present shortened versions of the abstracts. We'll make sure to mention the title, the journal, the first author, and the last author for each publication. Whether you're in the lab, on the bus, or cooking your meal, we hope you find this podcast helpful.
Hi there, I'm your host, Ellen Koch, for this episode of AMiNDR. Today, we'll be covering papers on methods to detect amyloid or tau protein in the brain. These are all published in June 2020, and this episode is a real mix of papers, some using PET imaging, some developing fluorescent and antibody probes, and some that go into other interesting ways to detect these proteins. If you're new to the show, we are a neuroscience podcast aimed at researchers, including graduate students and postdocs, who study neurodegenerative diseases. And currently, we're focusing on Alzheimer's disease. We want to keep you up to date on the latest papers that have come out and hopefully help your research. That being said, we cover all of the 80 papers from the month, separated into episodes based on topic. All of the papers are peer-reviewed, but since we are covering literally hundreds of papers every month, we don't have the time to go into the nitty-gritty details or make judgment calls on the quality of the papers themselves. But we provide you with a bibliography for each episode, so you can easily find the original papers to delve deeper into the studies. Check the show notes for more info. We are a new podcast, so we really appreciate the support that we've gotten so far. We just launched over this summer, and we are almost finished all of our episodes in our June series, covering the papers that came out in June 2020. And then we'll be moving on to the papers from August, as we decided to skip over July for this past month. Our team has grown a lot, and we're growing and growing every day, and I just want to extend a thank you out to all of the team for all of their hard work. Now, let's start talking about amyloid and tau detection in the brain. Our first three papers are on detecting amyloid in the brain using PET imaging in humans. The first one tests the performance of a quantitative approach to interpreting PET images. It's called Feasibility Study of a PET-Only Amyloid Quantification Method, a Comparison with Visual Interpretation. This one is by first author Shimokawa and last author Sasaki in the journal Annals of Nuclear Medicine. The authors here compared their quantitative evaluation of amyloid PET with traditional evaluation methods. The standard method refers to a physician reviewing the images, and they argue that this approach is subjective, while their quantitative method is objective. The study retrospectively analyzed amyloid PET data from the Japanese Alzheimer's Disease Neuroimaging Initiative. The quantitative evaluation used the adaptive template method and empirically PIB-prone region of interest. For each area, they determined the standardized uptake value ratio, which I'll refer to as SUVR. What they found was that the quantitative evaluation performed as well as subjective visual evaluation by physicians. So, seems like this technique may be a useful way to quantify amyloid PET. Now we move on to a paper that uses deep learning to look at amyloid beta levels. This paper is called Application of Deep Learning to Predict Standardized Uptake Value Ratio and Amyloid Status on 18F Florbetapir PET using ADNI data. First author is Reith, and last author is Zaharchuk, and this is published in the American Journal of Neuroradiology. These researchers wanted to use deep learning in an effort to automate the calculation of SUVR from PET imaging data. Remember, SUVR stands for Standardized Uptake Value Ratio. They trained two convolutional neural networks, ResNet50 and ResNet152, on over 2,500 18F Florbetapir PET scans that were separated into positive and negative cases based on the SUVR. About half of the cases in this dataset were amyloid positive. They achieved more accurate results than humans with their best trained network, a difference of 96% by the network compared to 88% by humans. Check out the paper for more details on this network. Next up, a paper looking at diagnosing AD in MS patients. Its title is Using Amyloid PET Imaging to Diagnose Alzheimer's Disease in Patients with Multiple Sclerosis. This is by Kolenko, first author, and last author Malhotra. Cognitive dysfunction is common in MS patients, but these patients can also be affected by a second cause of cognitive impairment, Alzheimer's disease. This can make it challenging to isolate the cause of dementia in these patients. This study explored using amyloid PET imaging to help with this problem. 3 MS patients with progressive cognitive impairment underwent amyloid PET imaging. 2 of them were diagnosed with AD based on the imaging results and given appropriate treatment. This seems like it could be a good strategy to determine the diagnosis and the avenue of treatment for MS patients that show signs of dementia. Okay, so the next 3 papers describe improved ways to detect amyloid beta in animal models using imaging or other methods. First, a paper on hybrid PET MRI imaging. It's called Hybrid PET MRI Enables High Spatial Resolution Quantitative Imaging of Amyloid Plaques in an Alzheimer's Disease Mouse Model. This one was published in the journal Scientific Reports by first author Frost and last author Lee. These researchers wanted to improve the spatial resolution of small animal PET imaging for monitoring AD pathology in mouse models. I personally think this is pretty high-tech. They combine PET and MRI imaging by adding a PET imaging insert into a small animal MRI instrument. They validated the measurements by comparing 18F florbetapir uptake in the 5X FAD mouse brain between PET MRI and another hybrid imaging setup, PET CT. In their AD model, they found elevated cortical and hippocampal uptake, as expected, and also found increased uptake in the thalamus. This indicates a possible role of the thalamus in AD pathology. This next paper also takes a look at detecting amyloid beta in mouse models. It's titled Feasibility of a Label-Free X-ray Method to Estimate Brain Amyloid Load in Small Animals. The journal is the Journal of Neuroscience Methods, and our first author is Dahal, last author Badano. This paper showcases a new method that uses small-angle X-ray scattering to assess amyloid load in the mouse brain. It measures the X-rays that are scattered from the beta sheets of amyloid plaques. They found that the amyloid load of the mouse brain could be estimated by using a characteristic scattering signal from various locations. One important benefit of this technique is it's a label-free way to estimate amyloid beta levels in the intact head. This one was a little out of my expertise, to be honest, so please check out the paper for all the details. The last paper in this section looks at amyloid beta in the retina. If you're interested in this topic, I hosted an episode back in August which covered some other papers on this topic of looking at the retina in AD, which we had sorted under unclassified detection methods. So this is episode 12. Now, on to the paper. It's titled Age-Specific Retinal and Cerebral Immunodetection of Amyloid Beta Plaques and Oligomers in a Rodent Model of Alzheimer's Disease. The first author for this one is Habiba, and the last author is Tayyebi, and published in the Journal of Alzheimer's Disease. So, as I learned when I hosted the Unclassified Diagnostic Methods episode, AD has been reported to cause pathology in the retina, including amyloid beta deposits. So this study looked at the relationship between brain and retinal A-beta deposition in the 5X FAD mouse model of AD. They stained retinas and brain tissue from these mice with Congo red, theoflavin T, and A-beta plaque-specific and A-beta oligomer-specific antibodies. The retinas had increased THT-specific amyloid fibrils and A-beta plaques, but staining with the anti-A-beta oligomer antibody showed a decrease in retinal A-beta oligomers that was age-dependent. Interestingly, in the neural tissue, A-beta oligomers were localized to neurons, in particular in late endosomes. So this means that A-beta oligomer accumulation in the retina is actually inversely correlated with A-beta plaques in the retina. This could lead to development of an early AD diagnostic test that measures A-beta levels in the eye. That's an interesting paper to end the section with. Before we jump into the next set of papers, let's take a quick break. Okay, we are back. For the next 6 papers I'm going to talk about today, these papers deal with developing probes for amyloid beta detection in various animal models. There are so many different models covered here, from nematodes to zebrafish to mice, and it goes to show that neurodegenerative diseases can be studied in so many different model systems. I myself have worked with a few different model animals in neuroscience research, fruit flies and mice mainly, and I got a little bit of experience observing experiments in zebrafish as well when I participated in the Neurophotonics Summer School at Laval University. I found working with zebrafish fascinating. It's so cool to work with animal models that are transparent. It definitely makes in vivo imaging so much easier. Getting into the papers now, we first have a paper developing a DNA Walker nanoprobe. DNAzyme-powered 3-dimensional DNA Walker nanoprobe for detection of amyloid beta peptide oligomer in living cells and in vivo. The journal is Analytical Chemistry. First author is Yin, and last author is Gao. Not sure how to pronounce that, but it's spelled G-A-O. So a DNA Walker is a type of nucleic acid nanomachine that works similarly to the proteins dynein and kinesin. This means it's able to walk along the track of DNA or other nucleic acids. This lab designed a DNA Walker probe for amyloid beta oligomer detection. As the title implies, it is powered by a deoxyribozyme or DNAzyme walking strand. When A-beta oligomers are present, the DNAzyme walking strand cleaves the substrate strand that is labeled with the fluorophore TAMRA and is modified on a gold nanoparticle surface. This results in a fluorescent signal that enables quantitative detection of amyloid beta oligomers in vitro in living cells and in vivo in mice. Check the paper for more info on these experiments, but this sounds like an interesting new approach to detecting amyloid beta oligomers. The next paper is on a different kind of nanoprobe developed in mice. It's called Ultra-Small T1-T2 Magnetic Resonance Multimodal Imaging Nanoprobes for the detection of beta amyloid aggregates in Alzheimer's disease mice. This is by first author Cai, C-A-I, and last author Li. And the journal is ACS Applied Materials and Interfaces. These authors wanted to develop a non-radioactive, sensitive probe that could detect amyloid beta plaque levels and their location. What they created has a long name, which I'm only going to say fully once. Phenothiazine derivative conjugated sub-5 nanometer ultra-small ferrite nanoprobes. So you can see why I only wanted to say it once. These were designed for T1-T2 magnetic resonance multimodal imaging. The abstract doesn't go into much more detail, but the researchers claim that the probe had high binding affinity for A-beta plaques and good R1 and R2 relaxivities. Now we have a paper using a new probe for another type of imaging, 2-photon imaging. PYR-PEG, a blood-brain barrier penetrating 2-photon imaging probe, selectively detects neuritic plaques, not tau aggregates. The authors are first author Choi and last author Cho, and this is also published in ACS Chemical Neuroscience. The authors of this paper made a probe for detecting A-beta using 2-photon imaging that can also cross the blood-brain barrier. It's called PYR-PEG and was administered in mice intravenously. They found this probe could detect neuritic plaques in the brain and olfactory bulb of an AD mouse model. It selectively targets A-beta rather than tau or alpha-synuclein aggregates. The blood-brain barrier penetrating quality of this probe is very interesting. This is a way that A-beta aggregates could be monitored in animal models or perhaps in humans as well in the future. Next up is a paper that developed environment-sensitive probes to follow amyloid aggregation. It's called Environment-Sensitive Probes for Illuminating Amyloid Aggregation in Vitro and in Zebrafish. Journal is ACS Sensors, with first author Fueyo Gonzalez and last author Herranz. These authors created a special type of solvatochromatic dyes, which, if you didn't know, are dyes that change color depending on the solvent used. Their dyes change color based on the 9-amino quinolamide moiety to detect early changes in amyloid fibrillation. They altered the quinolamide by placing a variety of amino acid groups at the 9 position of the quinolamide core. They imaged these probes using 1 or 2 photon microscopy, and out of all of the probes that they created, one of them could follow amyloid beta aggregation in real time very well in solution, including identifying the different types of oligomers. It seemed to also perform well in visualizing inclusion bodies in a zebrafish model which had neuronal damage induced by MPTP. However, a quick bit of research tells me that this is actually a Parkinson's model rather than AD, so it would be interesting to see if this could be used to detect amyloid plaques in an AD model too. Next up, rational design of a conformation-specific antibody for the quantification of A-beta oligomers. The first author of this paper is April, and the last author is Vendruscolo, and this was published in PNAS USA. These authors describe a 2-step rational design method that they use to develop an antibody probe for amyloid beta detection. First, they designed an initial panel of antibodies to bind to different epitopes of the protein sequence. They did this to determine regions that are exposed in oligomers, but not in fibrillar deposits. Then, for the next step, they made a second batch of antibodies that they specifically targeted to these regions. The abstract focused on this approach, which sounds like a great method to develop these kinds of antibodies. You'll have to check the paper itself for the details on their results, but they seem to have found an antibody that can be used to detect and quantify A-beta oligomers in vitro, in C. elegans, and in mouse hippocampus. The last paper in this section has a short and sweet abstract. It's called Binding Modes of Theoflavin T and Congo Red to the Fibril Structure of Amyloid Beta 1 to 42. Our first author is Frieg, last author is Golk, and the journal is Chemical Communications. So this paper uses something that I'm honestly not very familiar with, but I do find very fascinating, and that's molecular dynamics simulations and binding free energy calculations. They performed these to predict binding modes of fluorescent dyes to detect amyloid beta fibrils. They used these to predict binding modes of fluorescent dyes to detect amyloid beta fibrils. These dyes are the amyloid beta 1 to 42 fibril dyes, Theoflavin T and Congo Red. And they found that these probes bind to hydrophobic grooves oriented with the long axis parallel to the fibril axis. The authors state that this can provide a starting point in designing novel molecules for better diagnostic tools for AD. Okay, so amyloid beta seems to be the favorite protein currently studied in AD, but tau pathology is also a major component of AD research. So the last 2 papers today deal with detecting tau in AD using novel PET tracers. First up, we have a paper titled Clinical Evaluation of 18F JNJ 64326067, a novel candidate PET tracer for the detection of tau pathology in Alzheimer's disease. This one is by first author Schmidt, last author San Diego, and the journal is the European Journal of Nuclear Medicine and Molecular Imaging. These authors are following up on a compound they previously identified that had high affinity and selectivity for tau aggregates. The entire name, I already said it in the title, and it's J&J and a bunch of numbers, so I'll be referring to it as J&J in this abstract. They performed 18F J&J PET scans on patients with probable AD compared to healthy controls. The probable AD status was determined by 18F fluorobetapir PET imaging, a standard way to diagnose AD. Their results showed that 4 of their 5 AD patients had visually apparent retention of 18F J&J in relevant cortical areas. In temporal and frontal cortical regions, the 4 patients had elevated standard uptake value ratios compared to controls. This could be a promising tool for measuring tau aggregates in AD patients. Our final paper today is titled Tau PET Imaging with 18F PI2620 in Aging and Neurodegenerative Diseases. The journal is European Journal of Nuclear Medicine and Molecular Imaging, and this is by first author Mormino and last author Chin. This study also used the novel PET ligand, which is called 18F PI2620, to detect tau. They imaged this in healthy controls, A-beta positive participants with cognitive impairment, and in participants with semantic variant primary progressive aphasia. They found that 18F PI2620 was elevated in medial temporal lobe, posterior cingulate cortex, and lateral parietal cortex in the A-beta positive participants. Interestingly, small increases were seen with increasing age in these regions in the control group too. So overall, this may be another good tool for detecting tau aggregation. And that's it for our episode on detecting amyloid and tau in the brain. If this is a topic of interest to you, you should check out our recent Imaging Tools for Diagnosis episodes as well, hosted by Bhumika. These are episodes 28 and 30. Amyloid and tau pathology in AD are also covered in many of our June 2020 episodes, including some of the preclinical and novel drug development episodes. So I hope you found today's episode useful and accessible. Good luck on your research, and here's Sarah with a few last words. Goodbye!
That's it for this episode. A huge thank you to the team that is working on sorting, summarizing, and scripting these abstracts, as well as the operations behind AMiNDR. The music is from Journey of a Neurotransmitter by Anusha Kamesh, musician and fellow scientist, and now member of the AMiNDR team. You can find the original piece and her other music on SoundCloud under Anusha Kamesh or on her YouTube channel, AK Music. Interested in joining the team? Give us a shout! We can always use help with content development, podcast editing, advertising, and you can be part of a new and exciting venture. Reach us by email at aminderpodcast@gmail.com or follow us on Twitter. Oh, we're also on Facebook now. Don't forget to subscribe to our mailing list if you want access to the bibliography for each of our episodes. The references come with timestamps. Timestamps. So you can more easily locate the paper that caught your interest. Check our notes below for details on how to sign up. And very close to this, you'll also find a link to our feedback survey. Because, your feedback matters to us. So please, pretty please, let us know how we can make this podcast a better tool for you. And last but not least, thank you for tuning in with us. And on this note, we hope you found our podcast useful and accessible. Until next time.